All right, so let's do the first part of this plating protocol. And we're going to do just a single really big dilution of this mixture of cells. So we have to prepare what we need first of all. So I'm going to pop that back in there. We're going to grow the cells on YEPD. This is rich media that has all the kind of food that yeast needs. So I'm going to take a single plate and I'm going to label it. Oh, sorry, I have to get a pen. So if you were doing this in person, I'd suggest rather than writing your name on every plate, which takes a long time, I'd give you all a code letter. So in my code letter, I think it's just going to be A. So remember to write A on your plate. And in the protocol, I've suggested you use really kind of quite descriptive plates labeling system, so it helps to keep track of what the experiments are. But of course, if you were doing your own experiment, you might not want to write so much stuff. So you can use a coding system. You could just number all the plates or whatever. But yeah, for our purposes, let's use the kind of long names I've suggested in the protocol. So let me see. Our first part is here, one big dilution. Label your plate with your code letter and YEPD, that's the type of medium, and 1 times 10 to the minus 5, which represents the dilution we're going to do. Right, so I think I said in the introduction, but I'll say it again, the approximate density of the cells in the mixture is 10 to the 8 cells per milliliter, all right? So that's the density of these cells here. So we're going to do dilutions with the aim of getting 100 colonies on the plate. So we use the estimate to get 100 colonies. So we're going to use one of these bottles of sterile water. These contain exactly a litre. We know we made them exactly a litre because we weighed the glass bottles when they were dry first, noted the mass, filled them with slightly more than a litre of water, sterilised the whole thing in the autoclave, and then once it was all cool, we reduced the amount of water so there was exactly 1 kilogram of water, which is of course 1 litre. Right, so we have a sterile bottle of water. I'm going to put my mixture in a wrap here. In fact, I think I might make everything here. Now, to spread, when I've done our dilution, in order to spread the cells on the plate, we're going to use some glass beads, which are sterile. So take a tube of glass beads. Do you want to zoom up there? They're just sterilised dry glass beads, and we put a few on the surface of the plate. And by a few, it doesn't matter too much. I think you probably need at least 3 or 4. If you had 10 or 20, it wouldn't matter too much. Now this is a bit tricky, and it's one of these things you kind of practice. So just put a few on the surface of the plate like that. Put the lid back on to keep the plate sterile. Put the lid back on, so if you drop it, you don't get beads all over the floor or over the bench, which would be quite annoying.
And always important to do your labeling before putting the beads in.
Yes, yeah, exactly. So if you forgot to label your plate, you'd now have a bit of a problem because the beads are on there and you'd have to turn them around and they'll stick to the condensation in the lid and so on. So that's another point. Thank you, Jasmine. A lot of the time, people who are not used to labeling plates write the labels on the lid of the plate. And actually, we rarely, only when we're introducing cells to the plate do we really use them this way around. As you can see, the plates in their stacks are all the other way around with the medium on the top. So right on this side, and usually even when we're examining the plate, we're looking through the medium and looking at the colonies through that side. When you're incubating the plates, it's really important that they're this way around. If you do them the other way around, the plates will dry out. The air in the incubator dries them, evaporates the liquid from the medium. So, I mean, it looks like solid, but really it's almost all liquid, right? 99% of this is just liquid and it's immobilized in a gel. Right. I think this is going quite well. So, pipetting. Let's just check our protocol. So the plan is to pipette 10 microliters of this mixture. So we can work out how many cells that should be, right? If there's 10 to the 8 cells in a milliliter, then we're going to take out 10 microliters. That is 1/100th of a milliliter. So we're taking out 1 million cells, right? Approximately 10 to the 6 cells instead of 10 to the 8, because we take out 1/100th of that of 1 mil. That's 10 microliters. We're going to put that into our bottle of water, so there'll now be 1 million cells in this liter of water. And then we're going to mix that up really well so they're well suspended. We're going to take out 100 microliters. 100 microliters out of a liter, well, 1 mil out of a liter would be 1,000. So 100 microliters is another 10-fold less than that, so it's a 10,000th. So that will therefore, we're taking out 1/10,000th of that million cells, which is, wait, did I say that right? Yes, that's 100 cells. Excellent. So we're going to plate 100 cells approximately. We don't know the precise density, about 100 cells onto the plate. And for the other dilutions, I'm going to let you verify for yourselves what the expected number of cells are. Okay. Well, I might talk you through it a little bit. Right, we've got our beads on there. I've got my mixture. I've got my liter of water. Now the pipetting. So the first dilution step is 10 microliters. So we want to use a pipette with the appropriate range. These ones go to 20 microliters. So 10 is exactly in the middle of that range. It's the perfect pipette to use for this. So this might be a bit hard to see, but I'll use words to it. As it happens, it's already set to 10 microliters. So this one we can do fractions of a microliter. So that would be 9.8, and that's 10. So the red dial gives you fractions of microliters in this case, and this would go up to 20 microliters. Okay, so it's already set for 10, perfect. And this type, we use these yellow tips here, the small tips. Right, this mixture of cells that we're diluting, the cells, yeast aren't motile, they can't swim, and they're quite heavy, so they sink to the bottom, and they've formed a little plug. I'll just put it there so there's a bit of background, and you can zoom in on that. So what we need to do is mix them up really well, and this is important for all your dilution steps. When you've got your starting mixture, you need to mix it up. And if you've let a suspension of cells sit for more than a few seconds, you need to resuspend them. There are two ways to do that. You can use a vortex, which I've used in this case. If you sometimes it's not possible to vortex whatever your thing is. You can also suspend them by just pipetting up and down. I'll show you that later. Right, in this case we can vortex. So this instrument's set to automatic, so when you press on it, it mixes. So you can just do that. The lid's on the tube, so the cells aren't going anywhere, and that's absolutely fine. They're going to be evenly mixed now.
Do you want to put that back on the block so they can see? On the
Oh yeah,
rubber
sure.
block.
Yeah. So you can see that plug of cells that have sunk to the bottom is gone. They're all nicely suspended. Right. So this is a sterile rack of tips. I'm going to take the first one. It doesn't matter where. Some people are very OCD and like to work along a rows. Actually, that's quite helpful sometimes if you're doing lots of dilutions to keep track of where you are. But they're all the same, obviously. So you shouldn't be touching any of these tips. Every time you finish using it, close the lid to keep the tip sterile. Okay, now I've actually let this sit for a few seconds, so I'm going to very briefly put it back, give it a re-vortex. So carefully click the top off the tube, put your pipette tip in, pipette up, and close the tube again. I'm going to put it in my litre of water. So it would have been sensible to loosen the lid first, but actually it was loose enough for me to manage. I'm going to go carefully in, and then I'll blow out the last bit of medium. So this is really hard to describe in a video, but I'm going to show you, I guess. Okay, first of all, let me eject this tip. Okay, the contaminated tips go into these bins. Right, so these pipettas, which you've probably used before, there are two kind of resistances you reach, two detents. So the normal range is, you normally work it like this. So when you collect the liquid you want to collect, you first push down, put the tip into the liquid you want to collect, and then, in a controlled way, allow the spring to raise, and that will draw the liquid up. When you want to let the liquid out, there must be an active verb for that, and I've forgotten it. You go down not just to that first step, but to make sure all of the last drops of liquid are out. You can push further down, okay? It's important when you're collecting the liquid that you don't push all the way to the bottom, because then you'll be drawing more than 10 microliters, all right? So I'm going to put that there. Right, I'm going to put the lid on this. So vortexing this isn't going to work because it's so massive, so we can just invert it a few times. What did I say on the protocol? Probably, I just said a few times. Okay, so I think that is probably quite well mixed. Right, I'm going to draw out 100 microliters from this. So we can't use this P20 because it only goes up to 20. We can use this P200. That's perfect. So it takes 100 microliters, and amazingly, again, it's been already set for 100 microliters. So take another yellow tip. Again, I forgot to do. Okay, here's a good example. If it might be that I took my tip and this was really stuck, so I accidentally knocked the tip on something, like this bit of wall or anything, or even my hand. In a situation like that, the tip's now contaminated. It's not a big deal. I can just change the tip, make sure I have loosened the lid, and take another one. All right? So be careful if you contaminate your tip to change it.
Although maybe you should mix again, now that you've done that explanation.
Right, so all those cells will be sinking down. So I wasn't too worried because that's quite a lot, but yeah, why not? Let's give it another mix. Remember to loosen the lid. Okay, that's now the tip. Carefully cut out 100 microliters. And now this is going to go on our plate. All right, now you want to go quite close to the surface of the plate. You might want to steady the tip so that all of that volume, and blow out that last bit by pressing past the detent. I don't know if I'm pronouncing that word right. It sounds like it should be French. Right, this is again something that you might not have done before, and you'll want to practice it. It's getting the right kind of shaking motion to spread the beads. So the beads are just going to spread that liquid that I just put on there over the surface of the plate. These are quite fresh plates, so there's a bit of condensation on the lid, so they're quite wet plates. It will take a while for the liquid I just put on to completely be absorbed. So what you want to do is first make sure you've spread the liquid, the cell suspension, evenly. If it doesn't dry straight away, that's okay. You can let them sit for a while, come back to them. If you find that there's a bit of a puddle, then you can just shake again. Once all the surface of the plate is dry and the beads are dry, I'll show you how to tip them. Actually, I should show now because otherwise it's going to be confusing. I'm going to, I don't want to
Can
swirl
we just
this.
see it wet first?
Yeah.
You want to
Okay.
just open the lid.
Okay, so do you want to zoom in and try and see if the light shows? Yeah, that's good. That's great. Professional, excellent. So it's still quite wet. But I will tip them away. So I'm going to tip I invert the plate like this, and then all the beads, sometimes they might want sticking, so you can tap it down. The beads now fall onto the lid, so you can then lift the plate, which has got the cells on them, away from the lid, and then we can tip these into something. Hang on a minute. What I'm going to do is take another plate and use that. So you can use an empty petri dish to collect the beads, or a beaker, or anything convenient. Okay? So these are contaminated beads, so these will need to be autoclaved before they can be used again. All right, and then that's our plate ready to be incubated. Okay.